Hey everyone, welcome back to another lesson. So before we start this a bit complicated lesson, I would like to give you a general introduction about proteins. So proteins are functional molecules that have specific function in our body, and there are thousands and so many proteins we know of, and each one of them is quite big and has a specific function based on the sequence of amino acids. So when we make a protein using the DNA and an RNA that we will study in the future, when we create a protein, the sequence will give us the exact function and the way the protein is going to be folded inside our environment. So we talked about the fact that inside the cells, and in general in our body, the environment contains a lot of water molecule, and water molecules are polar, therefore they will want to create dipole-dipole or ion-dipole interactions within different parts of the protein. Proteins contain different R groups, and some R groups are hydrophobic and some R groups are hydrophilic. And the folding, the 3D folding of the actual amino acids sequence will create a specific protein in a specific shape. So every time we will have a specific sequence of amino acids, because of all the intermolecular interaction in the surrounding, the sequence of amino acids will want to collapse and create a specific shape. And every time, 99.999% of the times that we are going to have this specific sequence of amino acids, it will fold into the specific shape in space. This specific shape determines the function of the future protein. The specific shape will give us the function and the properties of this protein inside and in general in our cell. So because proteins can get quite confusing, biologists decided to divide the structure into 4 main hierarchy levels. The first one is called the primary protein structure. The primary protein structure is only the sequence of amino acids. That's it. So we said that the primary protein structure is the sequence of amino acids in the polypeptide chain. This primary protein structure will determine in the future the function of the protein. But right now, the primary would be just many different amino acid residues connected together via peptide bonds. And that's it. Each peptide bond and each amino acid can be abbreviated as 3 letters short word, because we don't want to write the name of the amino acid again and again and again. So we abbreviate it as the following. Phenylalanine, for example, is Phe. Leucine is LEU. Serine is SCR, for example, and cysteine is CYS. This is just a general example of the names of the amino acid. And in order, the primary structure is basically only the sequence of amino acid. Now the next step is the secondary protein structure. And this structure starting to get a bit complicated. So again, do you remember that we talked about in previous lessons about the intermolecular forces and how they interact with each other, and different molecules can form different shapes in space based on the intermolecular forces? So we are going to study in the future, and I already mentioned it a few times, that everything under a chemical environment always want to reach the most stable state. And when it comes to intermolecular forces between dipole-dipole and ion-dipole moments, we want to create the most interactions between the molecules. So I will give you an example because I know it's a bit complicated. If you have a protein, a sequence of proteins, the polypeptide will want to fold in such a way that the dipole-dipole R groups or backbone will want to create the most amount of interactions between the two and the most amount of bonds. And it will be the most stable state that you can reach in this reaction. Secondary protein structure is just a way to show that the primary structure, the sequence of protein, the sequence of amino acids, is fold in order to create the most amount of interactions within the backbone, not the other groups. So you will see that there are two main ways. There are way more ways for the secondary structure, but there are mainly two ways a protein can, amino acid sequence can fold in order to create the secondary structure. The secondary structure are the dipole-dipole interaction between the backbone, different backbones of the different amino acids inside the same sequence. So as you see here, we have something called beta-pleated sheet. And beta-pleated sheet is when you have one part of the amino acid sequence interacting and folding within the backbone of the other part via dipole-dipole bonds and hydrogen bonds, some hydrogen bonds and dipole-dipole bonds between the different backbones of different amino acids. Because we want to reach this stable state of having as many intermolecular forces as we can reach, the sequence of amino acid will fold in a following way. And this is one example of how it can fold. The other way we are talking about that it can fold is called the secondary alpha helix protein structure. Alpha helix protein structure is when the primary structure creates this weird helix. And again, the helix are just the backbone interacting with each other, and in order to reach the most thermodynamically stable state, they interact with each other and create this shape in space, because this shape will give us and give the polypeptide the opportunity to create as many intermolecular forces as it can, and it will reach the most stable state thermodynamically. We will study about thermodynamics in the future. Just remember for now that in order to reach the most stable state, we need to create the most interactions within each other. This is the most favorable state to reach. So the secondary protein structure is when the backbone of the different amino acids interact together and form different dipole-dipole interactions. Therefore, folding the specific sequence of amino acid into either polypeptide beta-pleated sheet or alpha helix interactions shape. Each one of the following helix contains 3.6 amino acids on average. It's about 3 to 4 amino acids. And this is a way that we can actually create a more thermodynamically favored state, because the surrounding is water and we want to reach as many intermolecular forces as we can. Therefore, the following molecules will be folded into the following, either this shape or the beta-pleated sheet shape, or many other shapes that we don't need to be aware of. These two are the main secondary protein structure. The next step is more interesting. The next step actually determines the actual 3D structure of the overall protein, and it finally gives the polypeptide sequence the function of the protein. So when we call something a protein, it means it has a function. When it calls something a polypeptide, it means it yet to have a function, but once it's actually fold into its finished product, into finished protein, it will have a function. So now the tertiary structure is when not only the backbone form intermolecular forces between different backbones of different amino acids and form specific places in the polypeptides to create either the alpha helix or the beta-pleated sheet. Now we have the interactions between one, the surrounding water environment, and two, among different R groups. So we need to explain the fact that the backbone itself, and we will study it in the future, is quite planar and linear, which means when you have a sequence of amino acids, for example in the secondary or primary structure, the backbone itself will be quite linear. And it's very stable, it won't be able to rotate much within itself. It will be quite stable. Therefore, when it comes to interaction between the different backbone shape, we'll be able to form two but very kind of specific shapes. It will be quite limited comparing to the 3D structures we can reach when it comes to the tertiary structure. The tertiary structure is dependent on the R groups and different R groups of the amino acids. Now we know there are so many different R groups, close to around 20 different R groups, and each one contains specific and unique properties such as hydrophilic side, hydrophobic, polarity, charge, negative charge, positive charge. These charges and R groups can interact with each other and interact with the watery surrounding as well. And as we said, unlike the backbone of the carbon backbone of the amino acids in the secondary structure, the R groups can actually, they have more ways to rotate and to be on the amino acid. So it will be more free to create more shapes. So while the carbon backbone, the backbone itself of the amino acid, is quite linear and strict and stable, the R groups can actually rotate around the amino acid and can form different interactions and more interactions with different molecules outside and ions outside the amino acids. So tertiary structure is when different types of R groups interact with each other and also interact with the watery surrounding that we have. Because we want to reach the most thermodynamically stable, which means the dipole-dipole and the ion-dipole interactions will be and want to reach a point when they are saturated to the most, you will have the folding of the protein where the proteins that have the amino acids that have R group that can interact with the water, that are hydrophilic, will be on the outside of the protein in order to create the most surface area and the most interactions with different water molecule. However, the amino acids that are more hydrophobic will be in the middle of the amino acids, in the middle of the protein, in order to not touch the different water molecules that are outside because they are hydrophobic. This overall hydrophilic and hydrophobic interactions and the overall intermolecular forces will give us a specific folding of the protein again and again and again, depends on which sequence of amino acids we have. So the same sequence of amino acids will be folded into the same shape in watery environment in the specific pH again and again and again to the same shape because of the intermolecular forces and because we want to reach a point that we have the most interaction and the most thermodynamically stable state. When it comes to folding of protein, the most thermodynamically stable state, and I'm going to say it a lot because it's very important, is the state that you have the most amount of intermolecular forces and dipole-dipole moments around you. Which means that at the end, the molecules that have R groups that can interact with the watery environment will be on the outside, and the molecules, the amino acids that are going to be the most hydrophobic are going to be in the inside. Therefore, the overall structure of amino acid, the sequence of amino acid will be folded. You will fold it in a specific shape every time. And the tertiary structure will determine the function of the protein, the function of the polypeptide. And a polypeptide with a specific function is called a protein. Now our last protein structure is the quaternary structure. Quaternary structure is when you have different polypeptides come together through specific static bonds, specific electrostatic bonds. They are not attached to each other through a covalent bonds, but they come and they attach next to each other within electrostatic bonds. And these electrostatic bonds are basically intermolecular forces. And the intermolecular forces can form this very stable quaternary protein, but this protein is built from different polypeptides that all come together in order to form and have a specific function. A very good example for a quaternary protein is hemoglobin, which is built from different proteins, and hemoglobin is the molecule that carries oxygen inside our blood. I know this video was a bit complicated, but only remember from the video is that you have 4 levels of folding of protein. The first one is the primary, where you have only the sequence of amino acids. The sequence of amino acids will determine the way that the protein will fold in the future. Protein will fold because of the intermolecular interactions. And because we want to reach the most stable state, we will always create the most amount of intermolecular forces and interactions. Therefore, you have the secondary protein structure, which is the most amount of intermolecular forces and interactions between the backbone itself and not yet the R groups. Because of the fact that the backbone is quite planar and stable and it doesn't rotate much, we have 2 specific ways to fold the specific sequence of proteins. Usually the secondary protein structure is within the polypeptide within a couple of dozens of amino acids, and it creates this either beta pleated sheet, which looks like this, and it is dipole-dipole interactions within the different backbone of different amino acids. And we have the alpha helix, which is usually a helical structure, which is again dipole-dipole interactions between different amino acids. We have it because we want to reach the most thermodynamically stable state, therefore the amino acid sequence will fold. The next one is the tertiary structure. It was the interaction between the R groups, the connection between the different R groups, and the interaction between different R groups within the water environment and among itself. We said that the R groups properties are very important because they will determine if the amino acids will be inside the molecule or outside the molecule. Then we talked about the fact that multiple polypeptides that come together form quaternary protein. And this is the last level of the hierarchy of proteins. I hope this video made sense. I know it was a bit complicated, but I will see you in the next video.